Good morning, everybody. I'm Dominic Hazard, working at INRAE, and I will present to you a study dealing with identification of QTL for body fat reserves in meat sheep. This study was carried out by Tiffany Master during her PhD. As you know, in ruminants, body fat reserves in adipose tissues are a major source of energy. Such component is essential for various biological functions, such as survival, reproduction, health, and productivity. During a productive cycle, body reserve dynamic is characterized by the amount of body reserve at each physiological stage, and also by changes in body reserve over time. These changes are due to mobilization or accretion of triglycerides present in adipose tissue and involve lipolysis and lipogenesis pathway. An issue for genetic selection could be to produce animals exhibited a greater management of body reserves, for instance by increasing body reserve accretion before mating, or by improving body reserve mobilization during periods of high energy requirements. Body reserve levels assessed through body condition score or ultrasound fat depth are lowly to highly heritable, while body reserve changes over time are lowly heritable. No study so far has investigated QTL associated with body fat reserve in productive use. Therefore, the aim of the study was to search for QTL associated with body reserve levels and body reserve changes in productive use. To carry out this study, we work with the Romieux raised on fed outdoors all along the year on rangelands at the experimental farm Lafage on the coast du Larzac plateau. Reproduction is planned to have 1 lambing per year. We measured body condition score at 8K physiological stages during 1 to 3 productive cycles using a scale from 1 to 5 adapted from the grid developed by Russell and collaborators. We also measure plasma concentration at 4K biomarker of fat and energy metabolism, including non-esterified fatty acids and beta-hydroxybutyrate, and they were measured at 3 physiological stages during 2 cycles. Animals were genotyped using Illumina ovine S&P 15 and 50K beat chip. Low density chip was imputed on the medium density chip. After S&P quality control, 44,000 SNPs were used for analysis. Phenotypes were corrected for known fixed effects and took into account repeated measurement. Single SNP analysis was performed. For body condition score, a total of 600 U's were phenotyped and genotyped, and for biomarker we used 500 U's. Concerning the results, on the first graph I reported body condition score throughout a complete productive cycle for primiparous U in blue and multiparous U in orange. In the breeding system of Lafage, body condition score range between 2.4 and 3, highest scores were observed at the beginning of pregnancy, and the lowest score were observed at weaning. A markedly BCS loss was observed during pregnancy and suckling, and a BCS gain was observed after weaning. For biomarkers, I reported on the second graph plasma concentration for non-esterified fatty acids and beta-hydroxybutyrate at 3 physiological stages: before lambing, during suckling, and at next mating. So you can see the differences between physiological stages, and the highest levels of biomarkers were observed during body reserve mobilization period. Concerning genetic parameters, heritabilities for body condition score and concentration of biomarkers were moderate, while heritabilities for body condition score changes and biomarker changes over time were low. Concerning QTL analysis, for body condition score, we found 31 QTL. Only 3 QTL were highly significant, above genome-wide threshold. 17 QTL were associated with body condition score levels, and 14 QTL with BCS changes over time. Each QTL explained less than 4% of variance. Among these QTL, we considered 5 main genomic regions on chromosomes 1, 3, 8, 9, and 11 due to highly significant SNP and or presence of interesting candidate genes in these regions. For biomarkers, we identified 23 QTL, and only one reached the genome-wide significant threshold. 16 QTL were associated with beta-hydroxybutyrate and 7 QTL with non-esterified fatty acids. 2 main genomic regions were found on chromosomes 1 and 12. On this slide, I represented Manhattan plots for these 5 most interesting regions associated with body condition score. On chromosome 1, the QTL associated with BCS at mid-pregnancy. On chromosome 3, the QTL associated with BCS at the end of suckling. On chromosome 8, the QTL associated with BCS at lambing. The same region was also associated on chromosome 8 with the BCS loss between lambing and suckling. For this trait, we also found a QTL on chromosome 9. And last, a QTL on chromosome 11 associated with BCS loss between pregnancy and weaning. In these 5 genomic regions, we found interesting candidate genes close to the QTL we identified. For instance, on chromosome 8, we found a gene encoding glutamate metabotropic receptor 1 involved in energy storage and energy expenditure. We also found a gene TRPS1 on chromosome 9 associated with growth. On chromosome 11, we found the ATP binding acid genes associated with the transport of lipids. And on chromosome 1, we found the gene encoding leptin receptor, widely described to be involved in obesity or back fat thickness phenotypes. And in this region, we also found close to the leptin receptor gene coding for the phosphoglucosamidase 1 associated with gluconeogenesis or glycolysis. In conclusion, we found several QTL associated with body fat reserve in productive use, with small effect, but several genomic regions show interesting candidate genes previously described to be associated with fatness, energy, or lipid metabolisms, and also lipid transport. These results will lead to a better knowledge of genetic determinism of body reserve dynamic in sheep. Present results could also be used to improve efficiency and resilience in sheep by improving use and accretion of fat reserve. Of course, further work is required to dissect more deeply the underlying genetic mechanism and identify specific metabolic pathway involved in the variability of body reserve mobilization and accretion. Last, I want to acknowledge all collaborators involved in this work, and also la Région Occitanie, INRAE, and the European Union for their financial support. Thank you very much for your attention.